Hello friends, my name is Bhavish Chanduri and you are the Soldier Pharmacy. Then today's topic of subject is pharmacognosy and phytochemistry of unit first. Then first introduction of pharmacognosy. Then pharmacognosy is means that pharmacons means drugs, means crude drugs. Then cognosis means study and knowledge. Then definition of pharmacognosy. Pharmacognosy is a branch of science that deals with the study of crude drugs which are obtained from natural sources or origin like plant, animal, mineral and marine sources. Then phytochemistry, then phyto related to plants, then chemistry related to chemical compounds. That means the chemical which are obtained from plants. Then plant metabolites are divided into two types, primary metabolites and secondary metabolites. First, primary metabolites are directly synthesis from plants. Then they are biosynthesized by primary metabolites. Then second point, they help in growth and development of plants. Then secondary metabolites, second points, they help to protect the plant from any diseases. Then third, they are present in all parts of plant. Then present specific part of plants. Example of primary metabolites, first is protein, enzyme, oils and carbohydrates. Then secondary metabolites, example alkaloids, oil, resin, volatiles and glycosides. Then scope of pharmacognosy. First is herbal medicines, second is cosmetics, third is industry, fourth is academics, fifth is food of drug inspector, then sixth is traditional system medicine. A drug is any substance that can be inhaled, injected, smoke, absorbed via path on the skin or dissolve under the tongue that causes a physiological change in the body. Then sources of drug. First plant sources, second animal sources, third mineral sources, fourth marine sources, fifth plant tissue culture. First is plant sources. First, plant sources is the oldest and longest source of drug. Then second, they has been used in the treatment of various diseases from the ancient time. Third, traditional system of medicine like Ayurveda, Siddha, homeopathic and Unani system are based on the use of plants. Then fourth, many of the plant products are having important therapeutic agents like alkaloids, glycosides, fluorosides, enzymes, then volatile oils, etc. Then fifth is example. Plant name and constituent. First is leaves, then digitalis, eucalyptus, then tobaccos. Then constituent digitoxin, digoxin, eucalyptus oil and nicotine. Then second, fruits or flower. Name is opium or vinca. Constituent morphine, codeine, heroin, amorphine, vincristine and vinbastine. Then third, fruits, in places and revolfia. Then constituent imetin and isoprolene. Then fourth, var. Name cinchona. Constituent quinine, anti-malaria. Then quinidine, anti-arrhythmia. Then second, animal sources. It is the second largest source of drug. Certain animal plant and animal product are used of drug. The major graphs of animal products used in medicine are hormones, enzyme, organs, bile acid. Then hormones. Thyroidism moduli, preparation of thyroid gland of sheep and pigs. It is given orally to treat patient suffering from thyroid insufficiency. Then pancreas is a source of insulin used in the treatment of diabetes. Then ephedrine is a hormone produced by adrenal medulla and used a nasoconstrictor drugs. Then oxytocin is a polypeptide hormones obtained from pituitary gland by cattle and pig. Then second, enzyme. Then pepsin is a pyroticolytic enzyme of glycine juice produced from fresh pig stomach. Then trypsin is a proteolytic enzyme prepared from extract of pancreas. It is used for the topical application of the treatment of round ulcer, etc. Then pancreatin is a preparation which contains enzyme of pancreas, which is used to treat digestion problem. It is preparation from big pancreas. Then animal extraction organs. Liver and stomach preparation and bile are the example of this group. Honey from honeybees, beeswax from bees, crude liver from shark, wool fat from sheep, carminic acid from cochineal, then venom from snake. Marine sources. Major part of Earth is covered with water bodies. Hence, the bioactive compounds, chemical constituents are also obtained from marine flora, plants, and fauna animals. Used for the treatment of many diseases. Then, classification of drug of marine sources. First is antimicrobial agent or antibiotics. First is cephalosporin, then histamicin. Second is antiviral components. Then Irae, Apocetal, Averona, and Averol. Then third is antiparasitic compounds. Then first is domoic acid, then alpha cyanic acid, then laminine, then benzamide F, then anti-cancer agent, then sinularin, then halitoxin, then asparagine, then aplidine. Then fifth, cardiovascular agent. First is octopamine, then tetramine, then saxitonin, then laminine. Then sixth, anti-inflammation or antiplasmodic agent, tetrodotoxin and memolite. Then fourth, plant tissue culture. Plant tissue culture is a technique that is used to grow plant cell, tissue or organs under the sterile condition on a nutrient culture medium. It is a wide used to produce of plant. It is technique of control production of usual secondary metabolites. Advantages of plant tissue culture. Production of extract coffee of plant to quickly produce nature plants. Production of genetically modified plants. Then production of disease-free plant. Plant tissue culture as a source of drug. Secondary metabolites, plant sources, type of culture. First is rhizophyne, plant sources, Reulfaria, then type of culture, suspended culture. Then vinblastine, plant sources, vinca, in type of culture, cell culture. Then secondary metabolites, third, mecadine, then plant source, tobacco, then type of culture, suspension culture. Then organized drug and unorganized drug. Then first is these are organ or plants and animal and are made up of cell and are having definite structure. Unorganized drug, these are obtained from parts of plant by some process of extraction and purification. Then second is these drugs are named as flower, seeds, fruits, bark, stem, etc. Then these drugs are named as extract, latex, juice, resin, etc. They are solid in nature, they are solid, semi-solid and liquid in nature. Then 4. Microscopically study are useful in quality control. Then chemical study are performed for quality control or evaluation. Then example of organized drugs, parts of plant and example. Leaves, eucalyptus, tobacco and digitalis, wood, sandalwoods, then bark, cinchona and cinnamon, then flower, vinca, clove, saffron. Then unorganized drug example, classification and example. Resin, asafoetida, then gum and mucilage, acacia, transnat, gum, etc. Then latex, opium, a flea. Then wax, beeswax. Then dried latex. A latex is a product that contains special secretory tissue. The tissue plant are concerned with the secretion of gum, resin, volatile oil, and other substance. A latex is a milky fluid which is usually exuded after the tissue injury and incision. It is usually a white aqueous suspension in which small particles of oil globules are suspended. This nature suspension of milky consistency may contain protein, alkaloids, starch, resin, gums, oil, tannins, sugar, etc. that coagulate on exposure to air. Example, opium, papain, etc. These pharmaceutical latex were collected, dried and therapeutically used. Dried juices. These juices are obtained from extraction or pressuring of vegetables and fruits. The insertions are made up of two respective parts of plant, collected and dried. Example, leaves, aloe, then stem, quinoa, then dried extract. An extract is a concentrated preparation of the drug which is obtained from the remaining active constituent of the drug with suitable solvent and evaporating the solvent to obtain a residual mass or powder. The extract were prepared by using water, alcoholic, hydroalcoholic solution. Example of dried extract: tea, gelatin, catechu, latex from acacia used for the
food addiction, etc. Gum and mucilage. Gum are the abnormal product of plant metabolites formed by injury of the plant and by a bark down of the cell wall. They are produced by process known as chromososis. They are translucent amorphous substance of plant hydrocolloids produced by plant. They are soluble in water and give a viscous sticky solution. They are insoluble in alcohol and the other organic solvent. Example: tragnacanth, gum, acacia and gummer gum, etc. Then mucilage. Mucilage are also plant products which are similar to gum but are generally the namely product of plant metabolites from within the cell. With water, it form a shiny mask but don't dissolve. Example of mucilage: isobagal, agar, fenugreek, then squil. In pharmacy, gum and mucilage are used as a binding and thickening agent. Then aloe resin. These are semi-solid extracts which are composed of resin with volatile oil. Olive resin are prepared from ginger, cloves, capsicum, turmeric, collodium, and cinnamon. They are used as a flavoring agent in perfumery. Some are used in medicine. Olive gum resin. It is a mixture of volatile oil, gum and resin that is released from some plants and cells. Example: Asafoetida and Myrrh.